The technique for determining the arrangement of the protein coding genes in a genome is called exome sequencing. Now, what if scientists could use exome sequencing to identify genes for cancer? The proceedings of the National Academy of Science discovered a gene mutation that causes susceptibility to triple-negative breast cancer. This type of breast cancer does not express the genes for estrogen receptor, progesterone receptor, or HER2.
The gene in question, FANCM, has been linked to an increase in susceptibility to breast cancer. Stage 1 breast cancer is when cells begin invading normal tissue space and is split between 1A and 1B, where the 1A breast cancer has no cancer in the lymph nodes, but has tumors that can be up to 2 centimeters long, while 1B breast cancer is when cancer in the lymph nodes begins to appear. Stage 2 breast cancer is also split between two stages, 2A and 2B, where 2A describes cancer spread into the lymph nodes accompanied by growing tumors that can grow up to 5 centimeters, while 2B is almost the same as 2A, but with larger tumors. Finally, Stage 3 is split into 3 stages, 3A, 3B, and 3C, that each describe progressively larger tumors and more cancer being spread into the lymph nodes, accompanied by physical determinants of breast cancer such as inflamed breasts and noticeable lumps.
The proceedings of the National Academy of Science used exome sequencing of families with a history of breast cancer in the genetically homogeneous Finnish population to identify recurrent alleles associated with cancer risk. After combining results from the 3 phases of genotyping in 2 independent series of breast cancer cases and controls, a significant association with breast cancer was found for an FANCM nonsense mutation. A nonsense mutation is a mutation in a sequence of DNA that results in a premature stop codon of 3 mRNA nucleotides, which causes the ribosome to stop protein assembly. This premature stop has a strong positive correlation with breast cancer risk in the Finnish population. The nonsense mutation C5101CT was found to be significantly more prominent in triple-negative breast cancer patients. The experiment concluded this with a p-value of 0.0002, which was less than their alpha value of 0.05. FANCM is a gene that determines whether an individual has Fanconi anemia, a genetic disease in which an individual has a defect in the protein FACM, or Fanconi anemia complementation M, that is responsible for DNA repair. This means that the body has difficulty correcting mutations that occur during DNA replication, which can lead to deficiency in red blood cell production and the potential formation of cancerous tumors. BRCA1 and BRCA2 are human genes that produce tumor suppressor proteins and are suppressed in individuals with Fanconi anemia. These proteins help repair damaged DNA and therefore play a role in ensuring the stability of the cell's genetic material. If a mutation occurs on either of these genes, they cannot repair DNA properly, which can lead to cancer. These genes are more likely to be altered if an individual has Fanconi anemia. Other genes involved in DNA repair can also cause this aggressive form of breast cancer. The discovery of these genes is important and vital for assessing an individual's risk of breast cancer and for early detection.
Pedigrees show a largely homogeneous population of Finnish people, which depicts a very controlled sample for cross-testing. Even though Fanconi anemia results in a higher susceptibility to cancer in general due to the suppression of the DNA-repairing proteins, the FANCM mutation leads to a significantly higher chance of breast cancer compared to other cancers in the affected population. In relation to our current topic in biology, we can deduce from the pedigrees provided by the research group that Fanconi anemia is autosomal recessive. You can conclude that it is recessive because it skips generations. It is distinguishable that it is not X-linked recessive because, if that were true, all the sons of homozygous recessive females would be exponentially more susceptible to breast cancer, and based on the pedigrees, this is false.
The nonsense mutation C5101CT occurs due to a deletion at the 15th exon of FANCM, resulting in a stop codon. For example, this image depicts a similar nonsense mutation that results in a stop codon, uracil, guanine, and adenine, creating a premature stop that, with FANCM, cannot be repaired and, when duplicated, creates mutant DNA that could result in an abnormal cell growth, also known as a tumor.